Hey, Chris. As far as I know, I don't think I've ever been ice sculpting before, but all right. Thanks to everyone for showing up. This should be a lot of fun. What we do in my group is we look at measuring and modeling animal behavior. But as Chris sort of got at a little bit, we're a little bit different than what a lot of other groups do when they're talking about measuring and modeling behavior. In particular, we're interested in looking at whole repertoires of behavior. So not just one particular small action that an animal does, but how does an animal go about its day and do all the various activities? And how is that ordering of those activities governed? And then from there, what our eventual goal is to then move back into the nervous system and into the underlying physiology of the animal and understand how these things are relating and eventually creating behavior as their final output. But in order to do that, this sort of requires one to have a more sort of fundamental understanding of, or maybe instead of fundamental, the better word is precise understanding of what one means when you actually say the word behavior. And how can we use that word behavior in a way that makes it a mathematically useful concept that allows us to make precise statements and precise predictions as we move forward. And part of that is being able to think about it as a language. How can we think about a mathematical language for behavior in a way that allows us to do interesting types of things? And for starters, let's start with an analogy to other fields in biology, because I think it gives a little bit of insight. So let's imagine you have a room full of geneticists. You have this room full of geneticists, and there's this DNA. One is, there's some gene that you're particularly interested in. Now, DNA is this remarkably complex object with structure at many different length scales, hydrogen bonding, methylation, all these things like that. But we know that if you measure a relatively low-dimensional representation of behavior, so measure these base pairs or accordingly amino acid sequence, or if you're really fancy, something like methylation sites, you can go and that can be the basis for starting all the arguments and the discussions we as scientists like to have. And similarly, comfortingly, if one lab measures GATC, the lab next door also should be able to measure GATC. And so you have the sense of reproducibility as well. Moving up a couple layers of complexity, we can then talk about neurons, right? So neurons are even more complex objects, but we know that we can measure things like a membrane potential or a firing rate, and it's not the whole story. It doesn't tell us everything that's going on, but it gives us a relatively low-dimensional picture of what those things are doing and gives us a way of talking about and discussing. Now, the measurements we might want to make might be difficult or even theoretically impossible, but we do know that sort of if we could ask for data, what the sort of data we would ask for. And then we could go and use that data to then have conversations. But if I were to show you this, what are the numbers we put on this? There are things going on, all sorts of length scales and time scales. And so this is an aggressive interaction between two Drosophila, in case you're curious. And the question then becomes, how can we go and have a mathematically rich description of what's going on here in a way that allows us to be reproducible across labs and have quantitatively precise conversations. And so what I'm going to talk about today are sort of at least our baby steps towards doing that and the consequences and the things one might do once you start to build these types of representations. All right. So just take a step back for a moment. There's been a couple, if you think about how people have traditionally measured behavior, and there's been sort of two main camps, and I'm grossly oversimplifying here, but I think it's sort of a useful description and way to talk about it. There's been sort of these what I call coarse measurements and descriptive measurements. So if you go to a traditional, like a psych or a neuroscience department, you traditionally see things sort of towards the coarse measurement side of things, where you put an animal within a restricted environment is one potential way of doing it. So it's pretty typically you put this rodent in a maze and you measure mouse turns left, mouse turns right. Or you can measure things like latency to enter a particular open arm on, say, an elevated plus maze. Alternatively, sort of on this side of the world also, you could measure some coarse statistic like a mean velocity or, say, the number of times crossing the center of a dish. And these types of measurements are great in that they allow you to have something that's high throughput and reproducible, but you're only getting a very small portion of what the animal has actually done. And you have to put in the thing that you're measuring in order to You have to know the thing you're trying to measure to start out with. Similarly, on the other side, sort of over here, we have sort of descriptive measurements. And this is more what you would see typically in like an evolutionary biology or an ethology lab. And the traditional sort of paradigm for this is you go out and you have some trained expert, usually a grad student or postdoc, and you say, all right, I've watched this animal for a while. I'm an expert. I know what this animal does. It does these 7 things. And I'm going to make a little check mark each time it does one of those 7 things. And so we can then sort of modern methods. This is, of course, horribly time-consuming. Modern methods have made this a little bit more feasible through some nice supervised machine learning algorithms. And that also allows for a more degree of consistency. But you still have this problem that you've defined that there are these 7 behaviors, not 6, not 8, and these 7 and not those 7. And so you still have this sort of ad hoc definition of the behaviors. So we have this problem of sort of what we'd like to do is have something sort of in between, where you can have something that's relatively high throughput and relatively and consistent, but at the same time have something where we can get a more full description of what the animal is doing and try and do that with as few sort of ad hoc assumptions as possible. And that's sort of the goal. And what I'm going to try and convince you guys of is that a good way of doing this and a way of thinking about behavior is through the idea of stereotype movements. And so what do I mean by a stereotype movement? So this is going to, again, sort of show my physicist background. So imagine this is the space of all possible postures that an animal could make. So this could be, for example, say the 2 Euler angles my arm makes as I move around. So I could make some straight line motions, some periodic ones, do some crazier ones, or even do stuff that fills up the whole space. If you actually watch what an animal does, it doesn't do this. What you see is something more along the lines of this. We have a bunch of behaviors which are done a lot of the time, maybe with some noise on top, and then a few things which are sort of random or moving between these stereotype trajectories. And so we're going to call these things stereotype behaviors. So these are motions that an animal does and consistently across a wide range of timescales. And so the goal then is then to take these behaviors and say each time you do this behavior, you put a dot over there. Each time you go around this loop, you put a dot there. And we can then look at sort of the metadynamics moving around. And that's going to be the type of representation that we're going to try and build. All right. Philosophizing aside, let's move to actually some real data and see sort of how these things play out. So this is sort of the workhorse for a lot of the stuff I'll be talking about in the first bit of the talk. And it's basically a fly in an existentialist hell. It's running around in this plate. There's basically a fly. You put it here. There's a cover plate on top. And there's a glass that's running around on glass, and there's a backlight beyond it, and there's this camera sort of following it around, filming at 100 hertz. And we can just go and just watch these guys. And the question then becomes, just from watching these guys, what can we say? What's the representation, what's the repertoire of what this animal is doing in this relatively unconstrained yet kind of boring environment? And just as sort of a side note for the machine learning folks in here, is that so the data here I'll show for the first bit is 59 male flies for an hour each. And so if we do the math of 59 times 60 times 60 times 100 hertz, that gets you about 21 million frames of data. And for the stuff I'll show towards the end of the talk, it's actually 1 billion frames of data we have to analyze. So sort of my group's version of pipetting, the boring stuff that takes up all your actual time but you never talk about in your talks, is the idea of how do you actually deal with these large data sets and moving things in and out of RAM and things like that. And so if anybody is curious about some of the details about this, I'd happily discuss them later over lunch or at some other point. So, all right, so here's the data. So how are we going to analyze this? So I'll go through sort of this original pipeline that we were talking about, that we developed, and sort of this relatively high-level view. So the first thing you want to have is to make sure that if the animal is doing, say, one behavior over here, it does this behavior over here. So you want to make sure it has translational and rotational invariance. So we do a step where we take the images and just align them like that. And from there, what we would like to do, so I come from a biomechanics background in my PhD, and so biomechanics, then what you'd like to do is be able to track all like the limbs and the wings and all these nice little things, so then you can go back and dock that onto a physical model of the animal. The problem with that is that we have this huge data set. And these legs, it might not quite be able to tell from back there, but these legs look an awful lot like the wing veins. And so from one overhead camera, or even from multiple side views, you get too many occlusions to be able to do this tracking perfectly. And we have data sets of 1 billion frames, so even if you have a tracker which is 99.9% good, 0.001 times 1 billion is not a small number. And so we have to have something which requires exactly 0 human involvement in terms of doing the tracking. And how we do this is we use the idea that our images themselves are the raw data. So what we do is we take the images and we do one nonlinearity, but for the most part you can think of it as we do the images and then we do a PCA directly on the images. And so this gives us our set of eigenflies over here, where you can view these as a set of postural eigenmodes. So this plus, which is basically an image compression. So this times a projection, plus this times a projection, plus this times a projection, on and on, goes back, you sum those up, and that equals an original image. And we can do some statistical permutation tests and fun things like this. And you get there's about 50 of these modes. So don't take that number too seriously, but that's how we sort of get the number of 50 that we're going to wind up using for the rest of the analyses here. And so then what we do is now that we have these modes, we can then create a low-dimensional time series. So instead of, say, measuring the wing angles or the leg angles, we measure the projections onto these. And these give us some things involving basically what timescales are going on and what parts of the body are being used. I wouldn't go so far as to try and interpret. Like, you might be able to imagine that this is like the wings opening and closing. But then when you get to looking something like this, it's a little more confusing. But these are postures, right? And so when we think of behavior, behavior isn't just a posture. It's also a dynamic. It's this, we think of standing, if you think about the behavior, say, of standing still, right? Standing still could be like this, or it could be like, it depends on the exact configurations don't necessarily matter. Really what you're talking about is the motion. And so to go from these postures to behavior, we pretend we're like birdsong people. And we go and we look at the spectral representation. So we do a wavelet transform, which for those of you who haven't seen those, they're basically a multi-scale, short time scale Fourier transform where we have time on this axis, frequency on this axis, amplitude out of the page. And this is for mode 1, mode 2, mode 3, and so on. And so at each point in time, we have 25 frequency channels per mode. So we have 50 modes, 25 frequency channels. So 25 times 50 gives us a 1250 dimensional feature vector, which describes what the fly is doing at any given point in time. So what parts of its body is it moving, and at what relative frequencies. So because I'm a theorist, the idea then is to do something sort of audacious and potentially stupid and go from this 1250 dimensional space and try and plop it down into 2 dimensions. And we're going to do this in a very particular way. So what we care about, so if you think about, and this is sort of a sidebar for people that have thought about dimensionality reduction before, is that if you think about pretty much any dimensionality reduction method that people tend to use, what they do is they say, I've got these 2 points in a high dimensional space. I'd like to put them in a low dimensional space such that I preserve the long distances, which is a way of saying I want to preserve the overall topology of the space that originally was there. But we're looking for stereotyped behaviors. And so when you preserve these long distances in these traditional dimensionality reduction approaches, and this includes PCA, multidimensional scaling, isomap, all your favorite ones you've probably used, is that you wind up warping all these local clusters. We actually care about those local clusters. Those are actually the things we're looking for. So we need to have a dimensionality reduction method which keeps those local clusters similar. So if I have 2 points here, say the pink and the green, they sort of get mapped together if their spectra are close. And then the red and the purple get mapped further apart if they're different, but I don't really care how far apart as long as they're not close. And this method, which was originally developed by Lawrence van der Matten and Jeff Hinton back in 2008. This is this thing called T-distributed stochastic neighbor embedding. And I'll just play this gratuitous movie here, because that's one of the lab's specialties. And so this is just a toy model of a bunch of points plopped down into a 3-dimensional space. And then this is actually running this algorithm. And what you can see is that you've generally preserved the topological relationships between everything. So like this is connected to that, that's connected to that, good, 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 good. But now you see things like this point and this point, which once were far apart, are now close together, but not so close together that they wind up intermeshing. So it's a way of preserving the local structure of the data set while cramming everything down into this lower dimensional space. All right. So if we do all that, we go through all of that stuff, and this is what we get in the data. So this is always, so this is fun. This is 21 million points of data, so it's cool to say. And this is, so these are the result of doing this embedding on our flies. So each one of these points is one point in time. And there's a couple of interesting features about this space that we observe. One is that, to sort of use the technical term, this is clumpy, right? It's not sort of isotropically spread throughout everything. And we can see this a little bit better if we take each of these points and replace it by a small Gaussian. And we get this here, which is a probability density function over this two-dimensional space. And you can really see, you get all these peaks and these valleys in between them. But the other observation we can see is that, remember, each of these points is one point in time. So we can then watch to see, OK, what happens as we watch the fly move around sort of this weird abstracted space. And if we do that, we see something pretty interesting. What we see is we see this pretty dramatic sort of sit-switch dynamic, where it'll sit in one spot in the space, so like here, then switch quickly, sit, switch, and so on. We can see this more formally by looking at a PDF of the speed in this two-dimensional space. And we get this pretty nice bimodal distribution, where these two peaks are spread across by 2 orders of magnitude. So you really have it just sort of sit one place for a while, then move quickly. Sit one place, move quickly. So let's put these things together. So we have this space with a bunch of peaks, and we have this sort of bistable dynamic, where it'll sort of sit one place, then move, sit one place, then move. So we can ask, let's pull a bunch of random sequence of movies out where the fly gets mapped to this particular part of space. And let's see what the fly is doing during those trajectories. And we see that the flies are all running with the same speed. OK, that's sort of cool. Let's figure a different one. Let's look over here. If we look in trick over here, we get the flies are all grooming their head, actually with a slight left asymmetry if you look carefully. Or you can pick this over here, and the flies are all grooming their left wing. And we'll come back to left wing grooming in just a moment. So we call this our behavioral space, where each one of these peaks correspond to an individual stereotype behavior. And then we can watch sort of the fly as it sort of zips around in this space and creates a dynamic. So here's sort of just a movie, so you guys get the sort of the picture of what the dynamics are. So here's the fly's position in the dish, the fly, and then the fly's position in behavioral space. And when it goes in one of these pause states, it's going to turn cyan. So it'll be really quick. This is real time, then it's going to slow down in a second. So you can sort of see it sort of zips between these different things, and then it has non-stereotyped regions where it goes between these various peaks. OK, so we have this behavioral space. Now, what on earth can we do with it, and why do we care? So there's a couple of different types of measurements that we can do with this. I'm going to focus on one of them and briefly talk about the other one, because we don't have time to talk about both. But one of this is sort of this phenotyping mechanism. So one way to see this is we can try and look at, so here's a very gross phenotypical difference, looking at male and female flies. So these are flies, same rearing conditions, same age, same genetic background, all these things. And what we see is that these are young flies, and we see big differences between these two areas of the space and between these two areas of the space predominantly. So perhaps, and this is locomotion, this is idle. So we see the young male flies are much more lazy. Fine, OK. This is something we didn't need this big of a hammer to actually do, but it's always nice, and people in the literature had noticed this before, but it's always nice to be able to reproduce this and show that you're not completely making stuff up. But we can try and be more subtle than that and actually find something more sort of, more subtle. And so we can focus in on this region over here. So this is sort of posterior and wingy sorts of things. And we can subtract the two distributions in that region from each other. So this is the female PDF minus the male PDF. And so if it's red, that means the females perform that behavior more. If it's blue, it means the males do. And these black lines are lines of statistical significance after all your favorite multiple hypothesis corrections and things like that. So focus in particular on these two here. And so this, you have one behavior here that the female tends to do a little bit more, and this one that the male tends to do a little bit more. So I'll put these two up there. There's a really small difference between the two. A relatively large audience. Usually I ask the audience if you could figure it out, but there's enough people that have seen my talk here before probably. So what you can notice is actually the males actually kick their legs out to the side, whereas the females here take the legs straight back. And we can show this by going back to those icons and show that, yes, in fact, there actually is a real difference between these two. So is this an ethologically relevant distinction? Probably not. Is it possibly an artifact from the fact that there's a thing on the top and that the female flies are bigger than the male flies? Quite possibly. But the point is that, and sort of how I like to put this a little pithily, is that it allows us to be surprised. Right? I didn't have to look at my data set and say, I wonder if the left wing, if the males and the females groom their left wing slightly differently. It just sort of naturally pops out as you go through. So we're able to be surprised by our data set in the same way that sort of you can be surprised by a forward genetic screen. And so that's sort of the type of thing that allows us to do. So now one could imagine doing this in a much more complicated scenarios where we actually are doing all sorts of manipulations, say mutations or neuronal manipulations. And I'll show a little bit of those types of things a little later. Another thing that we can do, and I'll only briefly mention this, is that we've also developed this. So this is great work of this great grad student, Ugna Klabati, where basically we can go and apply a lot of these things to multiple animals running around. And so this is during fly courtship. And we can, say, isolate different types of behavior that emerge. And so here's a sort of fun movie of simultaneously measuring the behavioral space dynamics of the two animals. So we're doing similar things like this as well now with other animals besides flies, but I'll get to that in a little bit. And we can also, from this, do fun things like measuring dynamics of social interactions. Say, for example, given that the male is within 3 millimeters of the female, what does the behavioral space look like? Or, for example, given that the male is in this quadrant with respect to the female, what does the behavioral dynamics look like? And we can then sort of try and piece back together these types of causalities and correlations, trying to understand actually what this interplay is between the two individuals moving around in this sort of unsupervised way. And other stuff that I won't have time to talk about, which I'm pretty excited about, is actually applying a lot of these things to evolutionary dynamics, where you can take different species of flies and then move little bits of the genome back and forth between one species to another species, some fancy regressions to try and figure out what are the genetic regions corresponding to behavioral differences. And this is work that I've been doing with David Stern's lab at Genelia Research Campus. And the other type of measurement, which I'm not going to talk as much about, but we have some recently published work on, and I'd be happy to talk about a little later, is looking at sort of these timescales and these transitions of behavior. So one of the things that we notice, and one of the things that's interesting that I mentioned at the beginning, is once we have a representation, we can then watch to see how it sort of moves around in the map. So we can look at the transitions between these different behaviors. And one of the things we notice is that you get this really long time scales that emerge, which are far longer than you would expect for any type of Markovian model, where you're only looking a little bit back in the past. So you get these long time scales that emerge, and that these actually are in relation to sort of a hierarchical structure of hidden states or latent states within the system. So that's something that if you're curious about, I happily answer questions about later at lunch, or send me an email. It's not like I'm so far away. And other sorts of things you can then measure, say, circadian rhythms, like here, flies wake up. So you get all this locomotion stuff, go to bed, sleep, and there's interesting dynamics between these various things, and we can compare how these dynamics change as we mutate a bunch of factors within. We can also look at some fun things in terms of fly aging over here, and we get some interesting results. For example, like that the male flies have a midlife crisis and the females don't. But that's a story for another day. OK, so for the last little bit of the talk, what I want to talk about over the last portion, I want to talk about this question of how are behaviors encoded. And so this is a neuroscience audience. And so I think this, in some sense, is one of the fundamental questions thinking about when we're trying to link behavior to neurons, is that the brain is this complicated object, right? Even in a fly, we're talking tens of thousands of neurons. And there's this large amount of behavioral output. And so the question is, how does all of this get translated down into movement and motion? So, of course, it's a massive question that one can't ask precise things about. So let's try and narrow it down a little bit. And can we focus on a particular general principle that allows us to say something? And one general principle that we see across animals is this one. Namely, between the giraffe weevil here and the giraffe is the end of necks. Right? You have this brain, which has a bunch of neurons. You have a neck, which has a smaller number of neurons. And then you go out to the motor outputs here, which have this wide variety of stuff that occurs. So can we focus in sort of on the neck region and look to see what's happening in the neural processing and the neural encoding in that area, and try to say something about the underlying sort of structure of the behavioral commands. And so how we're going to do this is, again, we're going to go back to Mr. Drosophila melanogaster and to focus on these descending interneurons. These are neurons which have cell bodies up in the brain, and they have axons that go down through the ventral nerve cord, which is the fly version of their spinal cord, effectively. And again, my physicist background is going to show up here. And the idea here is to try and think of this in terms of the idea that, in fact, what you have to do is the brain is effectively compressing the information. So it's encoding behavioral command information down into the channel, which are these descending interneurons, which then gets decoded downstream into movement. And so the beauty of the fly is we have all these sort of fun genetic tools. And so this is where, again, my friends from Genelia come in. So this is work that's largely been done in the labs of Gwyneth Card and David Stern. And what they've done is they've made about 300 lines. So for the genetics aficionados here, they're split 4 UAS lines, allowing us to drive the gene of our choice into a very small and sparse subset of neurons within the fly. And in particular, we've focused on these neurons over here, which have these cell bodies in the brain and then have axons that travel down through the ventral nerve cord. And the beauty, again, of working with flies is that we can use this red-shifted channel rhodopsin. So we're going to use these lines to drive channel rhodopsin, but we're going to use CS crimson, which is actually, it's a red-shifted channel rhodopsin. And why redshift is nice for flies is that their skin is their cuticle is transparent to red light. So you don't need to do any surgery or anything. The fly is just running around on a plate. You shine a red light, neuron, you see what happens. And so this is sort of the setup. You have a bunch of flies running around. We do 12 flies at once, and we just basically pulse this red light on or off. So it's on for 15 seconds, off for 45, so on. And this is a lot of work that I did with Jessica Candy to get all this up and running. And it's sort of also, as a sort of sidebar, it's a really fun control environment because our control flies are the exact same genetic flies. We just don't feed them retinol. So that means that the crimson channel no longer works. And so they're still sort of subject to the same red light coming on and off, but they don't have the neuron being excited. So that's sort of our control that we have for this. All right, so to show an example of the type of sort of behavioral specificity, let's start with this positive control case of this neuron called PIP10, which was noted in Barry Dixon's lab a while back to be an upstream regulator of courtship behavior. So the fly is sort of doing its own business. Red light turns on in a second, right now, I think. And it goes and it starts courting uncontrollably, as you can sort of see by the fact these sort of wings sort of keep on flapping. And then the light turns off. And in a second, it's still freaked out, but it's no longer doing the courtship behavior. So now we can do something where we didn't necessarily know the answer. So let's look at, I promise there won't be too many of these low contrast movie images here. But we see, for example, you have this line here, which has a cell body, a couple of cell bodies here, and a couple of cell bodies here. And what we're going to do is we're going to look at this behavioral map as a function of when the light turns on. So for example, so this is at 5 seconds before the light turns on. The light's going to turn on at t = 0. You can sort of see beforehand, sort of spread throughout everything. The light turns on, everything goes up here. And it'll stay that way until you get t equals 15 seconds. Then it'll spread out through the rest of the space. And you can sort of see this. So here's our control flies that I mentioned earlier. And sort of you don't get any change throughout the whole stimulus period. Whereas in the experimental flies, you definitely see this increased sort of prevalence of this region up here. So we've developed some tricks to be able to go and statistically isolate where in the map do you get this sort of increased behavior. And you can, for example, this region up here gets upregulated here. And you can sort of see this really nice signal here in the red, and then the blue here are the experimentals. This consistently is activated throughout all the 30 minutes of the filming section. Similarly, you also can notify which ones are getting down. This one is actually one of those comforting ones. This is the idle region. Because we're making the fly do something, it's not doing something. And it turns out this region up here is grooming its head. So this particular line is involved in head grooming. Or we can pick other types of lines. So here's 3 different lines, which have different parts of the space here. And you get sort of different speeds of running, from fast down to slower. Not only that, you get different types of temporal dynamics. So here's where the light turns on in each case. And you get different types of dynamics that emerge. And we get this whole type of space. So here's our behavioral space that we create. And again, this is 1 billion frames of data here. So that's sort of fun. And so this is sort of the overall sort of topology of the space. And each of these sort of red shaded regions is the excited region from one particular line. So we're able to tile almost the entire behavioral space with this collection of lines. And then we can go back and try and ask some interesting questions about it. So one thing we can do is ask, all right, let's focus in on a couple of the different sort of morphological regions of the ventral nerve cord. So here we have these leg neuromeres, a textulum which sort of connects up in the middle, then this flight neuropill over here. One of the interesting things we see is that almost all of the behaviors we see, with a couple of exceptions, are modulated primarily through this textulum. So if we ask basically, if you perform a behavior, or sorry, the other way around, if you hit a particular region of the ventral nerve cord, what types of behaviors are you likely to see? And so if you do this for the textulum, we see this wide variety of sort of fast-moving behaviors and central pattern type things that emerge. And so that's sort of one of the interesting things which sort of we can preliminarily say from a lot of these things, is that we can see these centralized control over a lot of these pattern-generated movements, which perhaps is not surprising because of all of the decades of work on central pattern generators. But at the same time, it's comforting to sort of observe that you actually can get this in this, and that you can actually see this evidence for the centralized control from the actual neurobiology. Another interesting thing we observe is, and this connects to this sort of ideas of latent states and things that I didn't talk about previously, is the fact that some of these behaviors are context dependent. So it depends, depending on what behavior I'm doing now, when the red light comes on, I'll do a different behavior on the other end. So here's this line here. This is a pretty sparse line. It's just one cell body on each side. And so you can ask, so there's 2 statistically significant regions that get excited by this particular line, this yellow region up here and this white region over here. And so what this density is what's the behavioral space density conditioned on me transitioning to this after the light turns on. And similarly, this is the behavioral space density conditioned on me transitioning to this. So you can sort of see going over here, which is sort of like slow leg movements, that you can see it's pretty widely spread throughout the behavioral space. Whereas if I happen to go here, I was doing this one thing here. And you can actually quantify this is a significant difference. We actually get that we have half a bit of information. And so it's possible of 1 bit of information, and this is significantly different than 0. So I should also note this is work that I've been doing with my grad student, Jerry. And one of the interesting things is, doing most things, you go to this guy. But if you happen to be doing something with your wings, you go to this behavior over here very precisely, which is this sort of wing extension. So the people I talk to that think about fly courtship are pretty excited about this, because you then have this context-specific type of behavioral modification. So the behavior you're doing now then can affect what the exact same neuron is doing when you excite it later. And then we can go through this throughout our whole collection of lines, and that we see about 25 lines have a statistically significant mutual information greater than 0. And so then we're trying to piece this thing together, and this is work that we're just doing now, trying to sort of understand sort of how all of these various lines are coming together, and maybe that how this context dependence might serve as sort of a modulating general principle for how the circuit works. And that's sort of as an advertisement for other work we're doing, is that this is sort of the type of sort of latent state dynamics that we emerge if we actually look at even just the freely behaving animal. We get basically that there's these long time scale dynamics that emerge from having different types of the body moving at different frequencies and at different time scales. And so these long time scales seem to be oriented in this hierarchical manner. So it's possible that this hierarchy that we measure in the just the straight up behavioral dynamics might actually be implemented in these sort of neural commands areas. And so that's something that we're investigating, and hopefully we'll go full circle across both of these. And sort of lastly, for the last couple minutes, we'll talk about a typical question that is one that's done a lot of work in flies. One usually gets this one. It doesn't work in mice. And so the point I want to make here is that sort of this part is very sort of system specific. So you have basically your animals, and you have to find a way of taking your videos or whatever your data is and turning it into a low dimensional time series. So it could be you do this fancy thing like PCA, like what we did, or you have motion tracker on your object, or you're just better computer vision person than I am, and you're actually able to track your animal. So from there, you can basically, but once you have those low dimensional time series, the rest of this is pretty generalizable. And you don't need to make too many choices from there on. And so let's just try and see what happens if we do in a couple of the organisms. So one of my colleagues over in Tom Jessel's lab was nice enough just to take some movies of mice and their own existentialist tell. And we get this type of space where you get walking over here, or my dog's favorite behavior over here. And we get this type of sort of the same type of organization that we were seeing before. And we have evidence, well, and so then so this is more recent work I've been doing with Jim Kwan in Robert Liu's lab, actually looking at sort of pairwise interactions between mice and prairie voles during home cage interactions. And we have some work now where we're able to do these types of measurements while they're wearing neurologgers. So we're getting LFP recordings while they're doing this and trying to correlate actually the neural data and the behavioral data as they're sort of running around and doing it. But this is sort of very much in progress. And we have some other evidence as well that this works in other animals, other mammals in particular. So we have this lady doing the hokey pokey. And in one of my favorite plots I've made as a scientist. For example, hand, whole self, in, out, handshake, doing the hokey pokey, and that's what it's all. This is, by the way, the results of a bet with my postdoctoral advisor. But of course that's incredibly silly. But the whole point is that we can basically the generality allows us to apply it to a bunch of different systems. So if we have the social interactions, which I mentioned briefly. We have some things with high throughput screening where we're trying to piece together sort of the genetic components of different aspects of behavior. There's some work that hopefully will be published pretty soon looking at multimodal analysis, so looking at sort of mouse ultrasonic vocalizations and being able to do the same types of analyses with these as well. And there's some recent work that I've just started with a couple collaborators over at the Marcus Autism Center over at Emory, using some of these things to look at sort of clinical diagnosis and human movements. And so this is some work, just as sort of an advertisement. This is some very new work that I've been doing with Shella Kilholtz and Jacob Billings over in BME. And you can sort of see we can apply these types of data to fMRI data, especially when in this particular data set we have a set of behaviors. And so we're trying to sort of build this type of repertoire. And really the goal right now is to be able to use these approaches as a precise way of mapping between brain and behavior over a wide sort of modality of systems. And with that, I'll thank sort of the nascent members of my group, point out that there's code available for most of the things here, thank the nice people that have funded me throughout the years, and of course all the great collaborators I've worked with on various of these projects. So thanks.
Got time for a couple of questions if anyone.
Yeah.
How robust is the method for individual animals versus large groups?
It is, so here are the spaces from all of the flies in our, so we can measure, I mean, part of it is just different flies are different. So if one fly is doing a particular behavior, it always gets mapped to the same part of the space. And the same has been true for most of the mouse stuff. And that's largely limited by image processing, but I guess, so by robustness, do you mean, does the same behavior get mapped to the same spot for multiple animals? Yes.
How much variance is there versus, could you pull out meaningful differences across individuals that don't actually just suffer by losing data from averaging across individuals?
Yeah, no, so you can. So we have done this particularly, and that's sort of the hope, particularly for the mouse and the vole data, because we're not going to have 2,000 flies to do that, or 2,000 mice to do that. So yeah, so we can measure sort of meaningful individual differences between them, and we have on a few occasions, yeah. So, yeah.
So the only way that you look at differences is visualization. So like you had the example of the male and female fly with the leg that has subtle difference. But what if it's more subtle that you can't really see?
So that goes back, right, so that's a great question. So that goes back to sort of what I showed at the bottom here. So we can then go back. So now that we've recognized, OK, there are these two differences. I wouldn't necessarily call it as a visualization change, because we actually are doing a statistical test to isolate what these regions are. But we then can go back to these postural modes, and we can see basically which parts of the fly are moving differently in those regions. And so these are actually standard deviations, not SEMs. Here. So we do get this really clear difference in the behavioral performance between these two very nearby regions in the space. Now, granted, I showed you the modes that had the biggest difference, but still, I think that's a fair thing to do for this particular case. Yeah, Lena. Sure.
I have two questions. One is how can you use identification online, and how can you use label data to sort of document what you?
So there's a couple of things. So I'll do the first question first. The first question is, I don't know necessarily how to do it yet. I think you couldn't do the training online. The training.
Let's say you train it
right.
and you just like have notification
Right. So if I had a student that was an expert in deep nets and things, what I would say is what you do is you train the data, and then you could probably reasonably expect to train a deep net to say, given this image, what is sort of the behavioral mapping I would expect between them. And so that's sort of, but that would be probably a whole PhD project. But I think that that's certainly doable long term. The second question, I'm sorry, was about how do you use labeled data. And so one way of doing this is so in particular, one type of labeled data you could use is triplet labeling. So labeling, are these two things closer to the same thing than those two things? Right. And so then if you use triplet data, you can actually use those triplets to inform your embedding, which is actually how a lot of sort of the fancy stuff that Google and other companies are doing right now. So you can, and these are sort of called semi-supervised approaches, where you use supervised data to then create this sort of embedding representation. So that could be one thing, and that would be a way of necessarily pulling out the, because with the idea then would be, because you have these labels and you would like to be able to pull out those labels more representatively. Yeah. That certainly would be a way.
You had a lot of 2D images of the behavioral space being right. So one question is, are there higher dimensions? And I'm sure there are.
Wait, you haven't even asked the question yet. You're coming up with
I've been asked this question many times.
But that's not actually my question.
OK.
My question is, how much of the difference
Okay, so the first bit of the question is that you actually, is it the 3D, it's not so clear that you're actually using the 3 dimensions of the embedding. So because you're based, and I could get into some of the details of why I think this is true, but basically most of the behavior is reasonably well, almost entirely is well described in 2D as in 3D. You basically get a couple of % difference change by going from 2D to 3D. And so if you I haven't actually measured that specific quantity you're asking about, but given that overall 2 to 3% of the difference is, you only get a 2 to 3% difference going from 2D to 3D, it would be very surprising if all of that, if say the difference between 2 particular classes used all of that. And I always test this out in every single space I make, at least that's small enough that I can actually do the computation. And it always turns out like this, where basically you only get a couple of % change. So my theory on this is basically has to do with postural facilitation, the fact you have to do intermediate behaviors. And since you have to do intermediate behaviors, this means that most behaviors are reasonably well described as a planar graph, which you can then plop down into 2 dimensions in a relatively straightforward manner.
I trust you picked up I'm more curious about the dimensions than, like, you get a rotation, is your embedding changing? So if I got that neuron or something, or is my embedding changing, or is how I'm utilizing the space? So you're looking at a 2-dimensional space. How do I know? Are you using this first? Are you using this
So I think, I think to make meaningful comparisons, you have to put everything on the same space, right? So you can only see what you see. And so if you put everything in the same space, you get a 2D space out. And so if you're going to make that comparison, then everything gets mapped onto the 2D space.
That 2D space was selected for one of them?
No, it was selected for all of them. So you train on all of the data. So you actually train, because again, so it could be some neuron excites some really weird behavior. Right. So you want to make sure that that weird behavior is in your space. And so we have to do all sorts of important sampling and things. What?
What if the neuron is changing? That means that, like, there's the muscle synergy stuff that I think you know as well, that like the representing patients had more, had a higher number of synergies, which I think this is not synergy.
Right. So again, this is more of this is a low-dimensional abstraction of the high-dimensional space. And since you've trained them all together, and all of these things fit in the 2-dimensional space, I think the most sensible way of talking about them is how the 2-dimensional space changes. So now, if it happened, that's sort of a separate, interesting question. This is not in the natural coordinate system of the animal, right? It could be that each of these places in space have very different types of synergies between all the postural eigenmodes that generate them, but that's why they got put in that one part in 2D space. So then you have to go back and figure out. So this is sort of then when the human comes back in at the end, is then try and deconstruct what's actually happening with the underlying dynamics.
Let's maybe take the rest of this offline, or if there are other questions. Before you move, before we thank Gordon one more time, there were 2 things that, as I was trying to think up the ice sculpting competition example, I neglected to mention. One is, I showed you the schedule for the spring, but this is actually not the final talk of the fall semester. We have one more talk next week. So be sure to come back next Monday at 11 o'clock. Oliver Hobart, HHMI investigator from Columbia University, will be here. Should be very interesting. C. elegans, but really much broader than that, focused more on connectomics sort of questions using C. elegans as a model system. So please show up for that. Second thing, I showed you a long list of exciting people. Half of them were flying in from all over the country. And we're providing lunch every week, and I've neglected to mention that is all due, again, to the generosity of the Neural Engineering Center that is providing the resources for this series to continue for the entire Georgia Tech community next semester. When you see the current directors, Garrett Stanley, Lena Ting, Robbie Terra, a former recent director until he moved up to the dean's office, thank them for providing you lunch every Monday and something intellectually engaging to come to every week. So thanks for your time. Let's thank Gordon one more time.